Okay, I am Jale from Ege University in Izmir. At moment, the temperature 35 degrees Celsius. Is okay. Now, our research focused on the conversion of wastes, every kind of wastes, animal wastes, industrial wastes, and lignocellulosic wastes into fuels and materials by thermochemical process. What we do, we produce biochar, we produce hydrogen, and we produce carbon absorbent and sensor. Now, today, I'm going to speak on the utilization of biochar as seed coating material. Of course, I'll give the results from lab scale experiments, not the previous presentation. Okay, but first, I would like to give brief information about plant growth promoting bacteria and seed coating, which are important for sustainable agriculture. Plant growth bacteria is able to colonize the root and is beneficial for plant growth and development. In future, they are expected to replace the chemical fertilizers and pesticides. These bacteria are already used as biofertilizers and biocontrol agents and are commercially available as dry powder, suspension in oil, and liquid forms. Besides PGBB, seed coating is also considered one of the best methods to promote sustainable agriculture. Seed coatings makes seed shape, size, weight, and seed surface more uniform, and to make them easier to plant. The active compounds that improve seed quality and protect different kinds of insect, pests, and fungal. The materials can be directly applied into soil with coated seed. A broad range of materials in the process of seed coating are used. And according to materials functions, these materials can be categorized into three groups: binders, fillers, and active ingredients. Binders and fillers are used to provide for thermal, physical, and mechanical characteristics of coated seeds. Generally, the binder used are natural or synthetic polymers, for instance, methylcellulose, carboxymethylcellulose, polyvinyl alcohol, and gum arabic. Fillers are popularly bulking inert materials and allow for physical modification. And generally they are peat, talc, bentonite, and chitosan. As known, the seed industry is moving toward the greener future, and the exploring microbial coating is one of the alternatives. Microbial seed coating is to reduce the use of agrochemicals in sustainable agriculture. And here in the microbial seed coating, the filler acts as carrier for microbial inoculum. In case of seed coating with plant growth promoting bacteria, the bacteria that support plant growth and plant defense enter the direct soil with the seed and consequently the resource vary. But the most important risk in practice is the short shelf life of PGBP-coated seeds. In our study, we investigate the usability of biochar as a bacterial carrier to extend the shelf life of PGBP-coated seeds. As you know, biochar has many benefits, and several studies have shown that biochar is an effective carrier for microbial inoculants due to its favorable properties that promote the microbial life and enable them to resist external environmental changes. With high porosity and specific surface area, biochar can provide living space for microorganisms. The immobilization of microbes under biochars includes attachment on the surface and porous through adsorption, electrostatic interaction, ion grid formation, and covalent binding. Recently, seed coating with biochar has received much attention due to its ability to benefit plant productivity and microbial inoculants. As seed coating material, biochar is desirable and cost-effective carbogenous substance. This manuscript reviewed currently published study on the roles of biochar in seed coating. In our study, we produce different kinds of biochars from different vestas using different carbonization methods, and also we produce activated carbon as a carrier material. We use the pine sawdust and waste-dried fig. You can ask why waste-dried fig. Waste-dried fig is a waste from a food industry and contains high amount of toxin. Because of this, they destroyed by burning. We use it because they contain high amount of nutrient, which can be food for microorganisms when they turn the biochar. And also the other aim to use for dried fig was the waste management. We obtained the biochar, it's different temperatures and different carbonization method. In dry carbonization, we use 350 and 600 degrees Celsius for as pyrolysis temperature. And the obtained biochar are defined following order: B, biomass type, and pyrolysis temperature. In the wet carbonization experiments, we used only pine sawdust as biomass feedstock. We also produced the activated carbon because, as you know, activated carbon has more porous and higher surface area than biochar. The activated carbon produced from sawdust by chemical activation. In our study, we investigated can they produce biochars and activated carbon can be suitable carrier for seed coating with selected PGB bacteria, namely gram-positive and gram-negative bacteria. This bacteria, Bacillus, is one of the most promising bacteria for plant growth promotion without harmful side effects. And it is already used as an excellent agent for biofertilizer and biocontrol in agriculture. Although most previous studies have reported that biochar can enhance soil microorganism communities, the effects of biochar on different microorganisms can be quite different. The composition and physicochemical properties of biochar derived from different biomasses have differences. Thus, their effects on organisms should be different. For instance, researchers observed an improvement of microbial growth when using poultry litter-dried biochar, whereas they observed an inhibition of the same microorganism community when using pine biochar. Biotoxicity is the critical to use biochar as microorganism carrier. For this reason, first we performed cyclotoxicity test with our biochars and activated carbon. This test is very simple test. First, biochar was extracted with water, and biochar water extract was added, bacteria culture incubated, and then for every 2 hours, 1 milliliter solution was drawn, and number of bacteria were determined by UV. If the curve is parallel to control curve, it indicates that the biochar is not toxic. And also, shell life testing. We carried out shell life testing. This test examines how long a bacteria remains safe and effective under certain storage conditions. In this test, the bacteria culture was prepared by incubation, and then added into saline water, and then mixed biochar and homogenized, and then dried. Then the dried mixture was placed in a plastic bags and stored at room temperature, because, as you know, in market the seeds stayed at room temperature. And monthly intervals for several months by plate count method was used. Just we count the white microorganism. Okay, now let me show our results. As expected, our carriers, biochars and activated carbons, have different properties, also depending on the pyrolysis temperature and also depending on feedstock type. The main physicochemical characteristic of biochar that could be suitable for microbial inhabitation and growth in soil includes high porosity, efficient water holding capacity, appropriate carbon content, functional groups, and nutrients. Also, I would like to note that the volatile matter. The volatile matter it was very high in case of hydrochar. The volatile matter is good or not for microorganisms. Sometimes it is good because the microorganisms use the volatile matter as food, but sometimes it's not good. Sometimes volatile matter contains toxic materials and lead to dying the microorganisms. The metal inorganics are very important for the agricultural use of biochar or biochars, also for microorganisms and bacteria. Elements such as calcium, potassium, magnesium provide nutrients directly to plants for plant growth. Zinc and copper support plant growth, but zinc sometimes is inhibitor of some enzymes. You don't know. Always depends on the seed type. In this, on this slide, you see some textural properties of biochars. Also for comparison purpose, the textural properties of chitosan. Chitosan is the commercial and generally used as carrier. Chitosan also given. You see the biochars, most of biochars have higher surface area and higher pore volume than chitosan commercial one. And also, it is interesting, the pore size is chitosan much higher than our carriers. The carbon stability allows biochar to remain in the soil for a long time. Labil carbon refers to unstable carbon. High stable carbon content is ideal for long-term carbon storage, as you know already. The amount of labil carbon is related to volatile matter, but interestingly, labil carbon is rapidly metabolized by microorganisms and provides short-term soil fertility. And the biochar obtained from wet carbonization is high amount labil carbon. Now is the toxicity. In the toxicity, you see depending on the biochar type and also bacteria type, we see some differences. For example, sawdust biochar obtained 350 degrees Celsius is toxic for gram-positive, but non-toxic for gram-negative. And also biochar obtained from wet carbonization, non-toxic for gram-positive bacteria, but toxic gram-negative bacteria. It's very complex. The toxicity of biochar is very complex. But the first, we thought that, okay, this is the biochar from sawdust here is toxic for gram-positive. We thought that, okay, maybe PAH content was high, and we determined the total PAH content of biochar. As you see, the total amount was lower than the limit. Of course, these limits, PAH limits, is for soil, but we don't have any standard for microorganisms, PAH limit, in case of PAH limit. And we still investigate why is the toxic this toxicity. This biochar shows toxicity for gram-positive bacteria. We continue investigate. The other biochar, remember, the obtained from wet carbonization, and that biochar was toxic for gram-negative bacteria. For this investigation, why the toxic, we washed our biochar with water, and after washing water, again we controlled toxicity and was non-toxic. And we can say that some compounds remain on the biochar led to toxicity because this wet carbonization, the carbonization takes place in aqueous solution in water. And also we analyze the processed water and washing water. What are there inside? And as you see, some phenols and hydroxymethylphenol and also phosphorus are inside. And we can say these compounds are toxic for gram-negative bacteria, but non-toxic for gram-positive bacteria. And also the last results are the cell life test. As you see, the initial number of bacteria here and the other columns per monthly numbers, shows numbers. Some bacteria, for example, this gram-negative bacteria is dead after 4 months storage. And also this activated carbon, if we use the activated carbon as carrier, gram-negative bacteria is dead after 4 months, and even after 3 months, the number is less than 3. As conclusion, we cannot, as I said before, the mechanism of microbial introduction with the biochar surface is yet to be not explored. Just we know that the gram-positive bacteria form spores. That means resistance to drying, low nutrition condition. Gram-negative bacteria do not form spores, so storage stability is lower. But still, we are trying to understanding the relationship between toxicity, short shelf life, and the physicochemical properties of biochars. And also, in the autumn, we start the seedling emergence test for biochar PGPB coated seeds. That was all But if you have any idea, any questions, welcome.
Thank you.
Okay?
Thank you very much, Yale. That's a very interesting piece of research there. You got one question from Jair or Laura. Um, is there any information on the genotoxicity of biochar or changes to the genetic information that may lead to malignancies? So I'm not maybe you understand what that means.
I cannot hear.
Do you have any information on the genotoxicity of biochar?
Cytotoxicity?
Yes.
Already genotoxicity, no. Just, I am chemist.
Yeah, me
Also,
too.
I'm working multidisciplinary, and our colleague from microbiology department just cytotoxicity test
Yeah.
and shell life test is possible.
So I have another question. So you checked the hydrochar
Yes,
after leaching.
yes.
Did you also check the low temperature wood biochar after leaching?
Yeah.
And
But as I said before, the dry carbonization the remains the volatiles in the organic materials, hydrophobic materials, not dissolved water. Because of this, we cannot wash. But the hydrochar, in case of hydrochar, the hydrochar obtained in aqueous phase. Because the remains on the hydrochar can be water soluble.
okay, so you didn't see any difference when you washed the dry biochar. Okay.
Yes.
And do you know, so it seems that the gram-negative bacteria is far less
Yes.
survivable. Do you have an idea why it works less with them?
My colleagues explained that my gram-positive bacteria form spores.
Okay.
Spores. That means resistance to drying, low nutrient condition. Gram-negative bacteria do not form spores. Is very kind bacteria.
Okay.
He
Okay.
explained so.
Okay. Yeah.
But later we are deeply investigating. We are deeply investigating.
and
Because
and
of this, I asked our hosts if they have an idea. Please let me know.
yeah, I mean, I guess it's quite novel, the whole concept. And because you want to seed coat, um, you want to use a biochar for seed coating. Do you have an idea how you would coat the seeds?
It's very easy. We will use the binder,
Uh-huh.
the binder, the gum, Arabic gum, and normally I didn't talk about it. The seed coating, three types: seed coating, dressing, film coating, and the pelletizing. But we will try very easy way, just film coating, using binder and film just. It's very easy. Just we plant everything for germination test, and in autumn we will start.
Okay. Great. So we will hear next year the results of
Yes,
the seed coating.
promise.
That's great. So we are a bit over time. So thank you very much for your presentation. And if there are any follow-up questions, you can write me or Yale directly. Yeah, and we look forward to your results next year.